rivers and coastal areas in different parts of the world. And then just finally, and this is my last example, is we're also starting to work on microplastics and how they affect the marine invertebrates of the Minas Basin. We've been looking at green crabs and blue mussels and eastern mud snails, but I'm only going to talk about blue mussels today. And what we did with our blue mussels is we put them, we collected them from the field, and then we compared the gut structure of field mussels from a lab control. So this is our lab control. Mussels that were exposed in the lab to low concentrations of microplastics, so 10 particles per cubic meter, or high concentrations of microplastics, so 1,000 particles per cubic meter. So if you remember that heat map of microplastics from Vancouver Island, we're way down in the color zone of that particular heat map. And these are histological sections of the gut of the control, the lab control, and the high treatment. And you do not need to be a histologist to recognize that these do not look the same. They look very different. And basically what we see with the microplastics, the cell structure is lost, the junctions between cells are lost, the cell height is lost, and the granules, the zymogen granules and calcium granules are lost as well. So the microplastics really can take a major toll on the structure of the gut. And Laura Ferguson, who was working on this project with me, is really interested in the microbiome. So she looked at the gut samples for changes in the microbiome. This is another cluster analysis. These are water samples, so don't worry about that. The orange and the red show gut samples, the microbiome of gut samples from mussels from the control and the low plastic. And so they're pretty similar. But when you compare that to the guts of the mussels exposed to the high plastic, it's a very, very different microbial community. And so they separate out in this cluster plot. And so this just shows you the different taxa that were detected in this microbiome analysis from the controls and the low microplastics. And these are the community breakdown for the mussels exposed to the high microplastics. And so some species really decreased in abundance and other species really increased in abundance. So the plastics, and who knows which came first, if it was the gut structure was lost and then so the microbiome was lost, or if it was because the microbiome deteriorated and so the gut structure was lost, because the two go hand in hand. But basically that was a story for the blue mussels. And so at ecologically relevant concentrations, and this shows a summary of some of the other studies we've been working on, the microplastics affected the gut structure, the microbiome of the gut. There's also effects of the gills on crabs. They really changed the immune response in the snails. And we have a little bit of developmental data from the snails that development is impacted as well. So this is just to sum up what I've said so far, the life and times of invertebrates of the Minas Basin. It's a really unusual ecosystem and we should not take it for granted. We should spend a lot more time out there listening and watching the organisms and finding out what their lives are all about. The high amount of suspended sediment is very critically important for the community as it is now. And of course, in ecology, things change, but a lot of those species are really well tuned into being exposed to high amounts of sediment. Some of them, an unusually high proportion, have weird examples of developmental plasticity, but this seems to work well for this very hypertidal, very weird environment. And separate really from the Minas Basin, but based on Minas Basin taxa, plastic leachates and microplastics affect these species at many different levels. And these effects can be manifest at really low concentrations. If you're not looking for them with specialized techniques like histology, you're not going to see them. And so it's really important to be trying to do these experiments at ecologically relevant concentrations, but with techniques that will allow you to see those changes. So for my acknowledgements, I'd just like to thank all of the people that have funded me in the last few years to be working on these projects. And that funding is greatly, greatly appreciated. And really, I want to acknowledge all the students that have worked with me in the last 5 years. They've been just wonderful in driving this research forward and coming up with wonderful, creative solutions to various problems. This is Sherman Blakeney, obviously not a student. I just put his picture in there. It's almost a year since he died. Oh, I'm gonna cry. No, I won't. I won't. He was my honor supervisor. Very close with him when he was still around. And I just wanted, I know some of you will remember him, so I just wanted to put his picture up there as well. Okay, so that's it for me. Are there any questions? I would love to have a chance to tell you anything I can about the invertebrates of the Minas Basin.
Thank you very much, Glynis.
You're welcome.
That was really interesting. Can you stop your screen share for a second so we can see who has questions? There are a couple of questions in the chat. So